Hello everybody and welcome to my oral presentation in bioengineering. Today I am going to talk about preparation and characterization of a gelatin-based coconut oil biomaterial and its effect on wound healing. My name is Mehlika Karamanlioglu and I am an assistant professor at Istanbul Gelişim University at Biomedical Engineering Department. This study is also conducted by my colleague Serap Yeşilkır-Baydar. Today I am going to talk about the background of my study. I will briefly introduce you about the methods that I used to conduct this study, which is mentioned in separate two parts. First one is characterization of films and second one is wound healing characterization of films. Then I will mention about the results regarding these methods and I will give you the general conclusions, and I also look forward for your questions at the end of the presentation. Biomaterials are obtained from renewable resources and they are applied widely in medicine and bioengineering fields and generally used in tissue regeneration purposes and generally as wound dressing materials. Biomaterials were determined to enhance wound healing because they are the analogues of protein and growth factors that stimulate healing of wounds. But we know that generally gelatin, which is a protein-based biomaterial, it's very much used in bioengineering and tissue engineering as well in different applications, as well as the wound dressing material. Also, coconut oil, which is conventionally used in food and cosmetics industry, it has also been used in medicine because in literature it's discovered that it has antimicrobial, anti-inflammatory, scar removal, and skin barrier effects. Therefore, the use of coconut oil has expanded from food and cosmetics industry to medicine as well. However, there is limited research on designing coconut oil as a component in a protein-based biomaterial wound dressing material, and also its effects hasn't been determined extensively. Therefore, in this study, we aim to firstly to prepare and characterize coconut oil incorporated homogeneous gelatin-based primary wound dressing materials for acute wounds. Also to investigate the cytotoxicity of this biomaterial that is synthesized from gelatin and coconut oil, and to look for the wound healing effects with a preliminary study. Our methods are basically divided into two, but first we prepared homogeneous uniform films from coconut oil and gelatin. Then we divided the methods into. First, we characterized films and we used some analysis in order to determine moisture content, water uptake, and water aging of these films. Then we determined the chemical structure of these films by using FTIR analysis, and then we did thermal analysis in order to determine the thermal properties of the synthesized films. The second part of the methods comprises of cell viability and wound healing characterization of films. Firstly, we used cell culture, and then on this cell culture we performed cytotoxicity assays, which is XCT assay, and then we did scratch assays to determine the wound healing properties of these films. Our first aim was to produce homogeneous films. Firstly, we did some control films that had gelatin and glycerol. First, we mixed gelatin and glycerol, and as you can see in here, we obtained transparent films. Then we added 10% coconut oil to this film forming solution, and you can see in here, you can observe the oil droplets, and you can see the non-uniform distribution of oil droplets in the second picture. Therefore, we didn't have the appropriate films for wound dressing applications. Therefore, we have to use an surfactant in order to uniformly disturb and emulsify these oil droplets. We tried various concentration of twin 80 as the surfactant. As you can see in here, this is the film that contains gelatin, glycerol, and coconut oil, and 1% twin 80. However, as you can see, still there is no uniformity. Therefore, we increased twin 80 percentages. In the D picture, in picture D, you can see we partly had uniformly disturbed oil droplets, but if you look closely, you can still see some oil droplets on the film surface. Therefore, in the last part, we tried to increase gelatin concentration to 20%. Therefore, we obtained uniformly disturbed, distributed oil droplets in picture E, as you can see in here. So, initially, we synthesized homogeneous films with coconut oil, which was 10% and 20% of gelatin films with plasticides with glycerol, and also the last picture had 3% of the surfactant twin 80 in the film forming solutions. We also produced control films with the same composition but without coconut oil. Therefore, in the further analysis that we are going to talk about, we are going to use these two films in order to observe only the effect of coconut oil in these films. In this composition of films, first we determined the moisture content of films, and we have found out that moisture content of films decreased when the films included coconut oil. Moisture content was about 48% when films did not contain coconut oil, but this decreased to 41% when the films included the hydrophobic content, which is coconut oil, to 41%. Also, when we analyzed the water uptake of these films, gelatin films without any coconut oil had more water uptake than the films that contained the hydrophobic content, that is coconut oil. During the third hour, which is the 180th minute, as you can see in here, the water uptake of the films increased to more than 300% when films did not contain any coconut oil, but when it contained coconut oil, it decreased to 200 degrees. However, after 3 hours, we could not determine water uptake of films that contained coconut oil because they were disintegrated in water. However, when it contained coconut oil, films first became saturated, and then their water uptake did not change, and then finally it started to disintegrate after 210 minutes. And in relation with this, we also determined water aging of films. As you can see in here, after 3 hours in water, control films without coconut oil just disintegrated. However, the films that contained coconut oil were still intact even after 3 hours in water. Therefore, films without coconut oil had more water aging percentage, which is 88%, and when films contained coconut oil, this decreased to 82%. And in literature, it is determined that lower water aging is preferred in wound dressing materials, which shows that when gelatin films contained coconut oil, it is more appropriate, it's more suitable to use as wound dressing material. Also, when we look at the chemical structure of the films, we observed protein bands of gelatin in both samples, and that is emit A band, but it shifted to lower frequency when contained coconut oil. And also its amplitude increased, showing that amine groups were involved in hydrogen bonding when films contained coconut oil. And you can see in here the position of the band decreased to lower frequency when films contain 10% coconut oil. Protein content and coconut oil content involved in bonding, and also this shows that amine groups of gelatin was involved in hydrogen bonding when films contained coconut oil. Wavelength and amplitude differences are reported to be related to interaction between functional groups of protein and the oil content. Therefore, we can see that both contents have been interacting structurally. We also observed some bands only when films contained coconut oil, as you can see in here. Pointed in circles. Carbonyl stretching vibration observed in 17 and 42, as you can see in here, only observed when films contained coconut oil. And also some other stretching vibrations were observed only when films contained coconut oil. Thermal properties of films also changed when films contained coconut oil. Melting peak at 25 degrees Celsius was only observed when films contained coconut oil. Also, there was an increase in glass transition temperature. As you can see, it was 56 degrees when films did not contain coconut oil, but it increased to 64 degrees when films contained coconut oil, and this was due to the lower moisture content of coconut oil containing films. Also, other endothermic peak observed after glass transition temperature, which is the melt-like transition temperature that disrupts the ordered structure of protein, also increased to 130 degrees to maximum at 160 degrees when films contained coconut oil, which also shows a higher molecular order of gelatin films when contained coconut oil, also with greater melting enthalpy. Also, there are broader endothermic peaks appearing in films when contained coconut oil, which also indicates thermal improvement of films. In the second part of our study, we did cell viability and wound healing assays. For this purpose, we used L929 cells, which had healthy and intact characteristics. XTT assay as a measure of cell viability results showed that films without coconut oil were toxic for cells at 100, 75 and 50 microgram per milliliter when compared with the 100% viable control cells, since these films seem to decrease 50% of the viability. However, when films contained coconut oil, films had proliferative effect on these cells at mostly every concentration, specifically at 5 microgram per milliliter to 75 microgram per milliliter. Therefore, we can say that coconut oil increased proliferative effect of these gelatin-based films. For scratch assay, migration ability of cells were observed at inverted microscopy to see if there is a closure of injury. As you can see in here, in the negative control that has either FPS nor coconut oil, we did not observe any closure of scratch representing the wound after 24 hours. However, when films contained coconut oil, we observed migration of cells after 24 hours, that is showing the closure of wound, that means healing after scratch assays. In conclusion, we obtained the suitable concentrations for uniform gelatin-based wound healing biomaterial, which is structurally and thermally improved without decreasing the water uptake. And In second part of our study, we showed the proliferative effect, increasing cell viability and wound healing of coconut oil. Therefore, coconut oil could efficiently be added to gelatin-based films for primary wound dressing material for acute wounds. I would like to thank my colleague, Assistant Professor Serap Yeşilkır Baydar and Istanbul Gelişim University Scientific Research Projects Coordination Unit, which funded this study. And thank you for listening to me. And if you have any questions, I appreciate them. Thank you.